Well, thank you very much for the invitation to virtually participate in your Life Sciences Tuesday event. KromaTiD is a small company based near Boulder, Colorado, and we certainly appreciate the opportunity to share with you some of our capabilities. At KromaTiD, we focus on direct measurement of genomic structure using our directional genomic hybridization, or DGH, platform. We work with many leading genome engineering companies to visualize genomic structural features from a few kilobases to the entire genome. DGH and sequencing measure different things and are complementary and orthogonal techniques. DGH is a single cell technique and captures structural and cellular context that is lost by analyses of pooled DNA that rely on bioinformatic reconstruction and prediction of structure. Sequencing is great for the analysis of edits at the base pair level and for measuring rates of off-target errors. Sequencing is not the right tool for determining heterogeneous, generally low levels of structural variance that result from misrepair of DNA double-strand breaks from any source. The right tool for that job is DGH. DGH directly measures structure and provides definitive visualization of structural outcomes. For targeted edit and integration site analysis, we have DGH Insight probes, but also offer unbiased discovery and detection of structural variation by de novo DGH assays, which we call DGH Screen. So where do DGH assays fit in within the landscape of genomic analytical options? The lower limit of detection for targeted DGH Insight probes is around 3 kilobases currently, although we are working toward even smaller targets. I will also mention that these targeted probes can be used for FISH applications as well. There's just a slightly different sample prep involved. Genome-wide DGH Screen provides high-resolution structural data as well as karyotype. Finally, there's G-banding with typical resolution of megabases. That is the current gold standard for structural analyses. However, we really are talking about orders of magnitude lower resolution than DGH. That said, KromaTiD does offer G-banding as an orthogonal analysis to DGH. Okay, so a quick overview of how DGH works before showing some applications data. With DGH, we analyze single-strand DNA with single-strand fluorescently labeled oligonucleotides. We design the oligos to be complementary only to unique target sequence, and our probes are pools of these labeled oligos. We do a sample preparation that results in metaphases comprised of 2 oppositely oriented parent DNA strands. The probes were engineered to only bind 1 strand, and this means rearrangements are very easy to detect. Any out-of-place signal anywhere in the genome corresponds to a rearrangement. In this fashion, DGH detects not only translocations and complex rearrangements, but also inverted regions. As I mentioned, we work with many of the leading editing and pharma companies developing gene therapies. Our custom assay services have been utilized by our clients all along the critical path to the clinic. We provide not only unique capabilities, but also bring complementary technical expertise to the table. We run a very process-driven operation under a quality management system, and that has enabled us to support multiple regulatory filings. So I'll spend the next few minutes showing some representative assay designs along the lines of what we do for our customers and with our collaborators. Most of what we do is very customized, but the idea here is to show how flexible these DGH tools are. So in these examples, I'm not focusing on the specific results. Rather, my intent is to show how we can parse the qualitative and quantitative information from DGH. In this study, we're using both DGH Screen and DGH Insight to evaluate edited iPSCs. In this design, there is a probe bracketing the expected insert site in 1 color and an insert-specific probe in a 2nd color. Now, this insert is 10 kilobase pairs, which would again be essentially impossible to do with conventional FISH. We're also using DGH Screen paints for assessing random misrepair events and essentially providing for kind of concurrent tracking of general genomic instability. As far as the process outcome goes, we measured the percentage of cells with 0, 1, and 2 on-target integrations. And we also observed an unexpectedly high level of off-target integrations. We can visualize the images in different fluorescence channels to make clear scoring calls. And we can parse the quantitative data many ways to suit the purposes of the study. Here we're showing the distribution of the number of off-target events. One of the potential applications for Insight probes is to confirm copy number at each integration site. So our development of this capability is ongoing, but it at least appears from relative fluorescence that many of the off-target integrations observed in these edited iPSCs are partial transgenes. As I said, we are working out approaches to qualifying this assay as a means of determining copy number, and there'll be more on that soon. I would next like to share with you some representative whole genome screening data. This study is underway as a collaboration with the U.S. government's National Institutes of Standards and Technology and involves Coriell cell line GM24385, which is the source of the famous genome in a bottle standard so commonly used to qualify sequencing techniques. This is among the most sequenced genomes in the world, and we performed whole genome DGH screen to characterize simple and complex structural variants, including traditionally difficult to measure inversions. Here we have profiled the distribution and prevalence of different types of variants by chromosome. Significantly, in this single whole genome DGH study, we not only confirmed the presence of a known inversion on chromosome 3, but also discovered a significant number of previously unknown recurrent inversions throughout the genome of the heterogeneous sample. The development of this whole genome screening method is being funded in part through small business grants from the U.S. government's National Institutes of Health, specifically through the National Center for Advances of Translational Sciences, as well as the National Human Genome Research Institute, which originally funded much of the sequencing of the human genome. Through this support, these NIH Centers of Excellence have acknowledged the value of a complementary orthogonal measurement of structure like DGH in applications ranging from cellular engineering, undiagnosed disease drivers, biodosimetry, and oncology. Hopefully, the above examples have shown the breadth and flexibility of the DGH technology. We are investing in further development, standardization, and productization of the platform, but I want to emphasize that we currently offer a range of commercial services based on robust DGH and pinpoint fish assays, methods, and processes. We're working with more than 20 companies right now and adding new customers all the time. Most of what we do these days is in the area of engineered cells, where we use our targeted and unbiased assays to confirm productive edits or integrations, as well as the extent of any unintended structural changes that have come about. Our customers include several global pharmas and many of the leading gene editing biotechs. During 2020, the CRISPR-based companies Editas Medicine and Intellia Therapeutics went public with KromaTiD data in publications and presentations, and other customers with alternative editing approaches are using our data to differentiate their platforms from CRISPR. There will be more publications this year. The large majority of the projects begin as custom services engagements, and we tend to fall in with therapeutic development programs as they progress through the pipeline with custom assays and ongoing research support. So far, a couple of these have led to regulatory filings containing some of our data. In response to this requirement from our customers, we have implemented quality management systems that support filing-ready data packages and are rolling out full GLP operating standards this year. So as a final slide, I will just kind of walk through how we get started on a project. Everything begins when a customer provides us target information and guidance on what they care about measuring. If a custom probe is required, we'll perform a design review and confirm suitability for our probes. We usually have at least one technical scope discussion where we flesh out exactly what the measurement objectives are, how many controls and active samples to include, and what level of sensitivity is required so we know how many cells to count. From this stage, we generally have enough information to generate a quote, and once signed, we provide a probe design description for approval by the customer, then trigger probe manufacturing. Once the project is underway, there is a fair bit of ongoing communication and structured project calls to put us all in the best position to get the project done right the first time. After we've looked at a representative fraction of the data from the samples, we generate a proposed scoring protocol and convene for the interim data review meeting with the customer. Out of this, a scoring SOP is generated for customer approval, then we finish up with the scoring. The deliverable is a report breaking out the findings as warranted by the scoring approach, as well as some representative images. We are proud of the fact that virtually every customer we've worked with over the years has come back for additional projects, and we've gotten a lot of positive feedback on this collaborative approach that we take. We are passionate about inventing ever better ways of measuring genomic structure, and we'd be pleased to discuss how we might help any of you who are interested. Thank you again for your interest.